The reason that we've selected vancomycin as the drug of choice in this particular study is this is a very commonly used antibiotic. It's one of the most commonly used antibiotics. And it's really quite complex to dose this particular drug optimally in patients. It's often used in the intensive care setting when patients have got dynamic changes in their physiology. And we need to get exposure of this antibiotic just right. And that's to ensure that we have enough on board or high enough exposure to kill the bacteria causing the infection, but not too much to cause toxicity. And what we're worried about, obviously, with vancomycin is acute kidney injury. Therapeutic drug monitoring of vancomycin is quite common around the world. So that's why it was sort of the first test case in this particular instance. In terms of how this particular sensor patch addresses a clinical need, for a very long time we've used trough concentrations to individualize therapy for drugs with a narrow therapeutic index, such as vancomycin, and it's quite a laborious process. We're always looking in the rear view mirror because we're collecting data from doses that have already been administered to the patient. And while we've progressed and some institutions have used AUC guided dosing using Bayesian software, adoption of that has been really rather limited due to various factors. So this technology, this real-time continuous sensing technology enables us to continuously monitor vancomycin concentration. So really simplifies the therapeutic drug monitoring process and ensures that we're treating our patients with the right dose of this important antibiotic and hopefully will improve patient outcomes. So I think the really exciting part of this particular study is it's the first time we've used this type of technology, so DNA aptamer sensing technology, to be able to monitor continuously in real time drug concentrations. We've been able to show previously in preclinical species, but this is the first time we've been able to show it in humans. We're monitoring just in the interstitial fluid, just on the skin, and we know that the interstitial fluid is a really good proxy for systemic drug concentrations. But it's obviously much more convenient given that it's less invasive than venous blood draws. So I think, first of all, is the initial use of this type of sensing technology, and also that paradigm shift in that we're sensing continuously in real time. I think the other key findings is just the level of insight that we receive from this type of technology. So we're monitoring concentrations every 5 minutes, and we've never seen this type of granularity in medical research before. So this is a level of detail that clinicians are potentially used to seeing, say, with blood pressure monitoring, but never with drug concentration monitoring. So for the first time, we're able to detect really dynamic changes in drug concentrations. And instead of estimating drug exposure, we can actually measure it. And that hasn't been possible previously. Then I think some other really exciting aspects of this study is just how sensitive it is. So really the sensor patch only reacts to vancomycin, which is obviously very good news. And then it's extremely accurate. So it's actually performs sensor to sensor variability, performs better than our current standard immunoassays. And certainly in comparison to spike solutions, it performs just as well as current immunoassays. So I think, to summarize, it's a really exciting study to be a part of. Shows for the first time continuous real-time monitoring of vancomycin using a technology which is extremely reliable. think in terms of acute kidney injury, so vancomycin associated kidney injury often happens because currently, we're only checking levels maybe once a day, at best twice a day in the intensive care setting. And even the AUC guided dosing, which relies on this Bayesian software, estimates exposure. So it doesn't measure it from these sparse blood samples. So this continuous real-time monitoring with the wearable patch essentially changes that completely, because you've got this information coming every 5 minutes. So clinicians can see very quickly when vancomycin exposure is starting to get too high, and they can already start either adjusting the dose very early on to prevent then acute kidney injury. So I anticipate that use of this type of technology will dramatically reduce acute kidney injury rates. In terms of other aspects, obviously, if you use this sensing technology, you don't need to do these specifically timed blood draws. That's a win not only for the clinical staff, but also for the patients, given that venous blood sampling is a relatively inconvenient procedure. So I think really it'll simplify that whole precision dosing process for patients receiving this important antibiotic.